Hello guys, today we are going to start with a very new topic and a very new subject, that is we'll start with the topic enzyme function and the subject will be biochemistry. For understanding the topic enzyme function, we should first know what is an enzyme. An enzyme, these are the proteins that play a major role in the biochemical reaction. So mainly the enzymes are the proteins that take part in the biochemical reaction. Enzymes also uses catalyst. What are catalysts? They help the process of the biochemical reaction to speed up. So the catalyst increases the speed of the reaction. That's why enzyme take the help of the catalyst. Every biochemical reaction has a substrate and a product. So we will, to understand the substrate and the product, we will plot it in the graph, and that graph is called reaction coordination graph. Our y-axis is the energy, our x-axis is the reaction progress. Here A we have taken as substrate and B is the product. Now the difference between the energy of the product, that is the energy of the product subtracted by the energy of the substrate, that is A, B minus A is equal to the energy of the reaction, also called the Gibbs free energy, that is the delta G. Therefore this distance is called as delta G, that is the difference between B and the difference minus the energy of the substrate. This was about the substrate and the product. Now the reaction occurs when the product has lower energy than the substrate. But here in this case delta G was positive because the energy of the product was higher than the energy of the substrate. But the reaction will only occur when the energy of the substrate is higher than the energy of the product. Therefore delta G will become negative. For again understanding the substrate and product work, we have taken again a reaction coordination graph. This is the y-axis, the x-axis, and here is the A is the substrate, the B is the product. Between the substrate and the product, there is a point called the transition point. The transition point has very high energy. So when whatever energy the substrate requires to go from A to the transition point is called the activation energy. This is called the activation energy. Once it reaches, the substrate reaches the transition state, it tries to either come back to the substrate or it can also go to the product. But what happens is the transition state is highly unstable, so it tries to find a very stable place, that may be either A, the substrate, or either B, that may be the product. But it will most probably will try to go to the product, because once it comes to the substrate, it will require the enough activation energy to go to the transition point. But once it becomes the product, it requires even more than enough activation energy to become the transition state, to become highly unstable. That's why millions of molecules going through this process, we get many of them as products. Now we'll study the substrate and enzyme. How does it work? Every enzyme is folded in a particular protein folds. That place is known as, this particular protein fold is known as the active site. All the substrates goes and binds to the active site. Then together they form the enzyme substrate complex. Enzyme decreases the extra energy required for reaction. What happens is, when this reaction was taking place without the enzyme, the transition point was here. But when the enzyme will get in, it will decrease the energy, so it has become a little lower than the previous one. That's why the enzyme decreases the extra energy required for the reaction. Now the classification of the enzyme. The classification of enzyme according to the IUB, that is the International Union of Biochemistry, they are divided into 6 groups: oxidoreductase, transferase, hydrolases, lysase, isomerase, and ligases. Some of their examples also I have written. These 6 classification we only need to remember. Then the enzyme kinetics. Enzyme kinetics is a very broad term and it's very difficult to understand, but we will try our best to understand it. First, let us know what is enzyme kinetics, the rate at which enzymes catalyze the biochemical reaction. The enzyme kinetics can be plotted in two ways. First one is the Michaelis-Menten plot and second one is Landwever-Burke plot. Let's understand the Michaelis-Menten plot. What happens here is that we plot a graph. The x-axis is the concentration of the substrate. The y-axis is the velocity of the reaction. The velocity of the reaction is directly proportional to the concentration of the substrate. Here we have written that. So when the number of enzyme is stable, this is the graph that we get. And when it is stable, we get the Vmax here. What is Vmax? Vmax is the maximum ability of an enzyme. That is the maximum ability an enzyme can work. So this point is the Vmax. Then what happens is that we need to find Km. Why? What is Km? The concentration of the substrate at which speed of the reaction is exactly half of the maximum velocity. So here the velocity is Vmax is exactly half, and we will go straight to this point. From here, this point will be on the concentration of the substrate. This point will be Km. We need to find the Km because Km is inversely proportional to the affinity, which means if Km is increasing, affinity will decrease. Therefore, so here we will find the difference as when the half of the Vmax is plotted on the graph, we find the Km. But this was the situation when the enzyme number was stable. It was not increasing, not decreasing. But when the enzyme number increases, the Vmax increases. When the enzyme number decreases, the Vmax decreases. This we need to remember. Enzyme and Vmax are directly proportional to each other. What is affinity? That we all know, but I'll say affinity is nothing but the binding capacity. Binding capacity. Then again it comes to the Vmax increase and decrease. For example, we can take when the Vmax increases is the induction of the gene expression. When the Vmax decreases is the repression of the gene expression. Under this, we get to know about two types of inhibition, that is non-competitive and competitive. Non-competitive inhibition. What happens here is that these are the two enzymes. This is enzyme 1, this is enzyme 2. What happens is substrate, this is the substrate, it will go and try to bind with the active site. The substrate binds active site of the enzyme. This is the substrate for this enzyme, and we need to remember that every enzyme has a specific substrate. But in the non-competitive inhibition, there is already a presence of inhibitory molecule. This inhibitory molecule will go and bind here with irreversibly to the active site, so that the substrate here cannot bind to the active site anymore. And this inhibitory molecule here will go and bind not to the active site but to the allosteric site, but this is reversible. So this process can take place again and again, and not letting the substrate to bind in the active site again. Due to the decrease in the substrate binding complex, therefore the enzyme will decrease, which will lead to the decrease of the Vmax. And in competitive inhibition, what happens is this is the enzyme. Here again the substrate will try to come and bind to the active site of the enzyme, but here is the competitive inhibitor, which will go and bind to the active site of the enzyme reversibly. But because it is reversible, but after binding it won't get metabolized, and as it is reversible, it will try to again go out and come back. This process will go on again and again. In this way the substrate will try to bind, but what happens is this will lead to the increase in the Km and decrease in the affinity. As we have already known, Km and affinity are inversely proportional. So this will lead to the increase in Km and decrease in the affinity. This was all about the Michaelis-Menten plot that we discussed, and the non-competitive and competitive inhibition. Now comes the second plot, that is the Lineweaver-Burke plot. This is also based on Michaelis-Menten plot, but here what happens is the initial speed is taken as V0, which is equal to the Vmax of the substrate divided by the Km plus of the substrate. But in Lineweaver-Burke plot, we reverse this equation, so it becomes 1 by V0 equal to Km of the substrate plus substrate by Vmax of the substrate. This is what happens in the Lineweaver-Burke plot. How do we understand this in the form of a plot? This is the y-axis, this is the x-axis. This is the plot, this is the line that we get in general, that is Km by Vmax, what we have studied here, the Km by Vx. This is in general. But when there is inhibition, there is a upgradation of the line, therefore it becomes 1 by Vmax, and when there is a stimulation, it degrades the line, therefore it's become minus 1 by Km. So the normal is this, but when there is inhibition or stimulation, these are the changes we find in the Lineweaver-Burke plot. So these were the two plots we also discussed. Enzyme kinetics is very broad topic. We need to read much more than this, but this was enough to get a general knowledge about the enzyme kinetics, and I hope you understood it. Thank you.